Hi all, welcome to Free Life Science class. Today we are going to study about some key concepts in translation. Okay, so let's discuss it. As an introduction, our translation is the process by which the polypeptide chain is synthesized according to the instruction in an mRNA. So from an mRNA, a DNA synthesize an mRNA. From the mRNA, a protein is synthesized. So this process of synthesizing RNA from DNA, then from RNA a protein is synthesized, and the whole process is known as central dogma, and we have been learning it from our childhood. Okay, so and the replication is the process by which a DNA is synthesized from another DNA. That is, DNA when duplicates or synthesizes for new daughter cell, we call this process as DNA replication. And an RNA is synthesized from DNA, we call it as transcription. Here the star indicates the reverse transcription process, that is from RNA a DNA is synthesized by the help of reverse transcription. And the next thing is RNA to protein, that is translation, and we are going to discuss this here. And the synthesis protein from mRNA is more complex because we know that our mRNA have four nucleotides, that is A, G, C and U. But when you compare it with the protein, you can see 20 different amino acids there, no nucleotides. So from nucleotides, it have to change to amino acid. So this is not at all simple task. It is little bit a risky task, but it is highly efficiently translated. And so that there are lots of components which are involved in this task, and we are going to discuss about that components which helps in this process. Do you know 80% of a cell's energy and 50% of the cell's dry weight is used up for the translation process? So you know how much energy does our translation mechanism require? How much energy? Need a lot of energy for this process, as it is a complex process. And here we need a lot of components which include mRNA, tRNA, lots of proteins, lots of enzymes which are involved in the process. Let us discuss about the components which are involved in this translation. And so the major components that are involved in the translation process are mRNA, tRNA, amino acid tRNA synthetase and ribosome, and all these components along with some other accessory proteins perform the task, that is translation. So here the here the first component of the synthesis is mRNA. And mRNA, you know that mRNA is the messenger RNA. This mRNA acts as template for the protein synthesis. Okay, it acts as template for protein synthesis. And if you compare any other process like transcription or a replication, where you can see DNA is acting as a template for synthesizing DNA itself. In replication, or DNA is acting as a template for RNA. Here the transcription is the process, but here in each of these processes, a nucleotide is synthesizing another nucleotide that is complementary to each other. But when you look into this, although the mRNA is the template, but the synthesizing protein have amino acid which is coded by triplet codon. Okay, triplet codon. So each triplet code for one amino acid. For example, AUG is a triplet codon which codes for methionine. Like that, we have 64 genetic codons which code for 20 different amino acids and 3 stop codons. Okay, so we have 64 genetic codons, and in that 20 are coding amino acid, 3 are stop codons like UAA, UGA and UAG. So these all are stop codons. And if we look into the mRNA, the protein coding region of the mRNA is composed of contiguous non-overlapping string of codons called the open reading frame. So if you look into an mRNA, you can see there are nucleotides like here I am writing like this. This is not actual nucleotides. I am just giving it for showing an example only. Okay. Okay, so here it is read like this. The first 3 as methionine, the next amino acid, the next one it is read as triplet codons, and each of these triplet codons is not overlapping. This G is not overlapping with this is not overlapping with this. It is a contiguous, that is adjacent triplet codons, which are read in a particular frame. Okay. Such frame is said to be open reading frame or otherwise known as ORF. Okay, so the next one is mRNA have the initiation codons like a start codon. So this mRNA have a start codon of AUG in prokaryote as well as in eukaryote. But if you look into prokaryotes, there are some other start codons like a GUG, then UUG, etc. act as start codon. Okay, so GUG or UUG act as start codon other than the AUG. AUG codes for methionine, GUG codes for alanine, like that. Okay, so what are the functions of the start codon? So for start codon, it specifies the first amino acid to be incorporated into the growing polypeptide chain. So a first amino acid to be incorporated will efficiently done with the start codon. So the start codon base pair with the P site of ribosome, where the first tRNA is present. First tRNA is present. So in the prokaryote, the first tRNA is known as F-met tRNA. We will discuss it later. And the second function of the start codon is it defines the reading frame for all subsequent codons. So first if we identify the first codon, then we can synthesize the other codons also into amino acid. That is, it will provide an open reading frame. Next thing about the mRNA is the mRNA also have another site where our small subunit of ribosome, that is 16S rRNA part, will comes and bind to the mRNA, and the binding site of this 16S rRNA is known as ribosome binding sequence, and it is otherwise known as Shine-Dalgarno sequence in prokaryotes or bacteria. Okay, so this Shine-Dalgarno sequence is also known as ribosome binding sequence, and it is just upstream of the start codon, about 3 to 9 base pair upstream of the start codon. So if you look into this, you can identify that there is a sequence just before the start codon, about 3 to 9 base pair upstream of the start codon, and where there is a base pairing between the 16S rRNA and this Shine-Dalgarno sequence. So the sequence present in the Shine-Dalgarno sequence is like this: 5'AGGA, GGA, like that. And this is complementary to the 16S rRNA, where you can say this like that. U C C U C C U, like that. So this is complementary to each other, so they will base pair each other. So this is giving a high efficiency in translation. So you can say that like this are ribosomal binding site increase the efficiency of the codon. So this is all about the mRNA. Now the second component is tRNA. The third component for this machinery is amino acyl tRNA synthetase, which will help to attach our amino acid to the tRNA. And the fourth one is the ribosome itself. So now we are going to discuss about the tRNA. So in the next video you will get more about the tRNA. So please subscribe to my channel and also click on the bell icon so that you will get notification when I upload new videos. Thank you for watching.